Let's go. Alright, here's our little overview. This is an okay studying slide, although there's a bit more information on the actual slides, and they're like pictures that explain what's going on. Up here at the top, by the way, we've got nevirapine, which is, there it is, boom, non-nucleotide reverse transcriptase inhibitor. And you've got something bound over here, but the actual nevirapine binding site is in this little pocket right back there. There we go. That little guy. And so I think that may be like an, I don't know what that is in this structure. We'd have to go look at it. But yeah, that's fine. That's not an Easter egg. Although, our Easter egg for today is this one. All right. So there is my keychain. And my keychain is a virus. Now, question is, can you tell me what kind of virus this is attempting to represent? So on the outside, I have, looks like a spherical ball, and it's got different kinds of proteins that are sort of made a little differently. And then on the inside, here we go, we've got an icosahedral shell. And if you open the icosahedral shell, you can see there's a little coiled up DNA. Now the coiled up DNA doesn't actually coil like that in the real thing, so we'll ignore that part. But yeah, what would be an example of a human virus that has an icosahedral shell on the inside, infects people, and has an envelope on the outside? Let's see, do we need a clue? I don't know that we need a clue for this one, because there are a couple of them, actually. And so that's part of the Easter egg, is tell me what kind of virus this could be. And then the thing that'll make sure that you've got the right one that I'm talking about, and we're talking about one that infects humans for sure, is that there's a special name for the proteins that fill in this space, sort of between the capsid and the membrane. And it's a word that starts with T and ends with T. And so, you're going to tell me what virus this could be, and anything, honestly, within the family would be fine by me. And you're going to tell me what's the name of this substance in between the capsid and the membrane. Yeah, so that's a weird Easter egg, isn't it? Yeah. And so, all right, go to town. Here we go. Okay, so these are all the ones. Look at them. Amazing. Yeah. And we'll go through these in order. And they're kind of arranged in roughly the order that they inhibit the virus. So we're going to start out with things that hit early in the viral replication cycle, and we're going to end with things that happen late in the viral replication cycle. Yeah, what can I say? It's very nicely organized. And at the end, I grabbed a webpage about current antiviral therapy cocktails that are approved, and so we can go look at those, just for information. Because I think one of the questions is for you to design an antiviral cocktail therapy. And yeah, we'll look at how people do that, and what they're trying to do, what the goals are, and yeah, there you go. Then you can do it. Okay. So, first step. At the level of entry, so this would be after receptor binding. Now there are drugs that have been in development that would prevent GP120 interaction with CD4, and they're going to try to bind in that little pocket where phenylalanine 43 is. And you, also would have co-receptor blocking drugs, which we will mention those in just a minute. But in addition to that, there are these class of drugs that are going to be similar to chloroquine. So an example of that would be hydroxychloroquine is one, and they generally have oquin at the end. Yeah, these things tend to work by inhibiting acidification of the vesicles. And so this would work at the level of GP41. So after receptor binding, as the virus is coming into the cell, it's being pulled inside, this would prevent that proton pump, the vacuolar ATPase, from putting H+ ions into the endosome. And so the early endosome would never mature to a late endosome because the pH would never really drop properly. And that pH drop is, of course, the signal for GP41 to change conformations and deploy the fusion peptide and reel in the cell and all, yeah, for all of GP41 to work. And so these drugs can have an effect there, but it requires really high concentrations of the drug. And these concentrations are often not achievable with non-toxic levels of therapy. So it's a thing that can work fine in a petri dish and in general does not work particularly well at the level of like an organism. So yeah. So you will see things like this tried out, and the papers usually don't go past saying, hey, works well in the lab. Yeah. And who knows, maybe it could work in a mouse, but probably not very well. The other drawback with this is that you're going to have a selectivity problem because this is how the cell eats. So you are interfering with the acidification of vesicles in every single cell in the body, not just the very few cells that are actually infected with the virus. And so you're giving every cell in the body some kind of eating disorder, basically. And that's not going to be great. So if you get enough in there to actually have an effect on the virus, it is likely to have an effect on the cells as well, which we would call a side effect. And we don't want drugs with very strong side effects. So yeah, this exists. I'm telling you it exists. But I'm telling you that at least as currently made, these are not effective antivirals. But our example would be hydroxychloroquine, and there is no resistance to this because the resistance would have to be at the level of the host, and yeah, you just kill the host. You poison the host as you're getting rid of the virus. Low-level poisoning. All right. So next one will be. And so this has a couple of names. Enfuvirtide is the one that tends to get used the most. Enfuvirtide. Other things you'll see it called T20. So like down here, there's T20. Or Fusion was one of the marketing names that those whiz kids in marketing came up with. And this is a fusion inhibitor. So after GP41 unfolds, after the fusion peptide hooks into the cell, this is an inhibitor, just like we talked about, of HR1 and HR2 coming together. So it's directed at GP41. It is literally a copy of HR2 from the virus. And sometimes they'll make a tiny tweak in the amino acids, but if you look at this thing, that's a very unusual looking drug because you notice it's got a peptide backbone to it. And it's got all these little side chains as if it was a big long amino acid, which it literally is. Yeah. So, has a narrow window of activity, but is still reasonably effective. And it hits the virus in a different way than some other antivirals. And so a good way to have synergistic effects is to push different enzymes in the virus at the same time. And by doing that, you can end up with a nice strategy that will knock a virus off that peak and push it down toward the valley. Yeah. All right. So resistance is found. Resistance occurs at the level of both HR1 and HR2 would change because HR1 and 2 start out separate, and then they're going to come together to make the 6-helix bundle, which here's our little 6-helix bundle from the side. And here, this yellow thing is the enfuvirtide bound to the HR1s. And so you can change HR1, but you've also got to change HR2 at the same time so that they still stick together. But now it's like you've changed the secret password for the clubhouse, and so Fusion doesn't get in. It turns out that the virus can mutate, can become resistant, but it pays a pretty high price in terms of fitness for these changes. And so that's why this drug, even though it's got a crazy small window of opportunity where it can be effective, it is still a pretty effective drug. Now, the big knock on this thing was that when it first came out, one, it's too big to actually ever be orally bioavailable, meaning you're going to have this through an IV drip straight into the bloodstream, or you're going to have no chance. Thankfully, the cells that HIV infects, those white blood cells, are all going to be located floating around the bloodstream or in organs where the bloodstream serves. And so, yeah, that's a good way to get the drug where it needs to be. Since it's a great big protein and not a little tiny molecule, it used to be super expensive. It was around $100,000 a year per patient when it first came out, and there's no way your insurance or mine is paying for that. But now they've got it down under $100 a dose. You go in there, you sit with an IV. I do not remember how long a dose lasts. I'm not sure if it's a couple of weeks. But yeah, essentially, it's used. It's actually used, even though it's kind of crazy in drug development terms. All right, next one, Maraviroc. You remember this one? This is the same slide from before. Yeah, I bet you do. So Maraviroc is going to bind down, and it's going to act directly on CCR5. It's going to bind in the bottom of CCR5, which is going to prevent binding of CCL5. So, yeah, you just knocked out your CCR5s. But it's also going to prevent binding of the V3 loop of HIV. And so, it's effective, but it's effective by basically taking out one of your genes. So you're it's like you're burning your own crops to deny them to the enemy, sort of a scorched earth defense. But it can work, and it's fairly effective as a complementary therapy. So the virus actually can become resistant to this. And with these CCR5 inhibitors, you will see that they can modify the V3 loop, and they're modifying it in a way where the V3 loop actually binds a little better than the Maraviroc, and so it can actually come in and occasionally displace a Maraviroc. But you also get changes in the region that the V3 loop is attached to, which is one of the constant regions. And let's look back and see which one it is. I want to say it's the C4. It is the C4. There we go. So V3 loop and C4, yeah. And you will get mutations in both of those that can be associated with some degree of resistance. But in general, it is harder for the virus to get real solid, knockout level resistance to Maraviroc, which is why it's a good antiviral. Yeah. Even though it's working at the level of the host, it's taking away something that the virus needs more than you need it. And that's the theory of antivirals in a nutshell. The virus has to do a bunch of things. Every step of the virus life cycle, every reaction that's carried out by viral proteins is potentially something that can be used against the virus, because as long as it's something the virus needs and the host does not need, you can pound on whatever that function is, and you can have a good antiviral effect. Yeah, all right. So, Maraviroc, not too bad, not too shabby. Okay, we'll do reverse transcriptase inhibitors, and then we will, carry on. So these are some examples of what we call nucleoside or nucleoside analog reverse transcriptase inhibitors. You'll see these sometimes abbreviated in the literature as NRTI, nucleoside reverse transcriptase inhibitors. I prefer to say nucleoside analogs because they're close, but they're not exactly like the nucleosides that make the nucleotides that make you and me have RNA and DNA. So for each of these, you'll see here's the natural molecule. So this is thymidine, and natural in the sense of this is the one that your cell is using right now to make some DNA. And it's DNA because it's lacking the oxygen right there. It is a 2' deoxy because that's position 1, that's position 2. So position 2 is where we're missing the oxygen. It's still got the 3' hydroxyl where we add stuff on, and then 4, 5, that's the 5' hydroxyl. So the 5' end is going to be hooked there, the 3' end is going to be hooked down there, and that's your 5' and your 3'. And so one of these drugs, and this is the one that we're going to cover in detail, is going to be AZT, which is azithromycin, which is just like thymidine. If you cover over this part that I'm running my mouse over, it's the same thing. Instead of the 3' hydroxyl, though, it's going to have an azide group, which is 3 nitrogens hooked up in a particular way. And yeah, that azide group is tough to displace, and essentially it becomes a blocker. And so what happens is that the HIV polymerase is searching through the cell looking for a nucleotide. It's looking for, say it's got an A there and it needs to put in a T, and so it's going to grab for a T. Let's say it grabs an AZT instead of a T. It's going to put in an AZT, but then when it goes to add the next nucleotide, there's no 3' hydroxyl there. And so there can be no next nucleotide, which means that, yeah, this is doing a thing called chain termination. So the chain is the piece of DNA that HIV is making, and if you can't add any more nucleotides after the drug is incorporated, then that is a chain termination event. And so that is one way that these drugs can work. And if you look at them all, so here, these are the normal nucleotides. These are the RNA versions over here. And you can see that there's our 3' hydroxyl. And then look at the antiviral. See? No 3'. 3', no 3'. No 3'. So it doesn't even have something where a 3' could be. No 3'. There's the natural molecule. And tenofovir over here doesn't even have a ring. Doesn't even have a proper ribose ring compared to ATP in a particular conformation. So it looks like the top of it, which means it can bind to polymerases. But yeah, once that's incorporated, you cannot continue the chain. The chain is terminated. And so you might ask, well, why do these not just kill all of our cells? Because our cells are made of DNA and copy themselves and make RNA, and if we just start terminating all of our chains, we'll also be dead. Yeah, and you're correct. There are potentially some side effects of this from exactly the mechanism that you say, but the cellular versions of polymerases tend to be a lot more careful. They have narrower, more tightly fitting active sites, and so generally an AZT won't fit in the active site of one of your enzymes, but it does fit in reverse transcriptase because reverse transcriptase is it's a little more open active site, and it's just not as careful, honestly. HIV is about going fast. It's not about being careful. Yeah. So that's a major way that these drugs can work, by chain termination. Another way is they can bind directly to reverse transcriptase and just sit there. And if they bind better than a nucleotide, then they will just occupy the active site. The enzyme isn't able to do anything, and you've just jammed the enzyme. Yeah. And that's fine too. That's another way that we can potentially win, and we're all about winning here. All right, so the thing with a nucleoside is that it cannot be incorporated directly because, of course, we don't put in just adenosine. We put in adenosine triphosphate, ATP. We only put in the triphosphate versions of these nucleotides, as you probably remember from cell biology, which means that when you put in a nucleoside, reverse transcriptase inhibitor, first it has to get charged up. So normally, if you were making a T nucleotide, thymidine, you'd start out with the little nucleotide with no phosphates. You'd have a particular enzyme called a cellular thymidine kinase that's going to add the first phosphate on there, and then you're going to have other cellular thymidine kinases or general nucleotide kinases that'll take you from 1 phosphate up to 3 phosphates, and now it's charged up, and now you can incorporate it. And when you incorporate it, of course, 2 of those phosphates go away, and the last phosphate remains as the little bridge between the nucleotide you just added and the previous 3' end, because we always add to the 3' end. Yeah. Okay, AZT, same process. And so we're going to use a particular thymidine kinase, a DNA thymidine kinase from the cell, and that's going to add a phosphate on. And then we're going to have a different thymidine monophosphate kinase, deoxythymidine monophosphate kinase. That's going to add the second one. And then you're going to have a just a nucleotide diphosphate kinase, which is going to add the third phosphate to one that already has two phosphates. And so boom, boom, boom. Now, for a nucleoside reverse transcriptase inhibitor to work, it has to not only bind really well to reverse transcriptase, really poorly to cellular DNA and RNA polymerases, it also has to bind well enough to deoxythymidine kinase, deoxythymidine monophosphate kinase, and the nucleotide diphosphate kinases so that it can be fully charged up and so that it can actually get turned into the triphosphate. You can make the triphosphate and try to put that in. Problem is, as the triphosphate is being incorporated into the cell, as it's being pulled into the cell membrane, it tends to lose those extra phosphates. And so it goes down to a monophosphate or an unphosphorylated version. And you'd basically have to start over anyway. Yeah, the phosphates are easily broken off. We have lots of enzymes that do that. So yeah, that's your problem. And so AZT works really well. It's a reverse transcriptase inhibitor. It binds to reverse transcriptase, and it's a chain terminator as well. But it causes fairly serious toxicity. This was kind of the first real antiviral that got traction and was used. And back in the day, it used to be used as a monotherapy. You would just get AZT and nothing else because that's all we had. And they'd try different doses and different things to make it not as bad, but it had some pretty severe side effects. And so it's good that we have so many more options now. Generally, at least in Europe and America, you will not see AZT used. I believe it is still used some places because it works. It's just that there are better things out there that work with fewer side effects or work a little better. All right, resistance profile. Because remember, resistance is the key to telling if something is really an antiviral. And yeah, you get resistance to AZT. Resistance comes in the form of changes around the reverse transcriptase. Let's look at them. So this is unfortunately switched around. So the thumb in yellow is this thumb in green, which is kind of pointed up towards you in this picture. So we've got the enzyme backwards. So here's the DNA strand. There's our DNA strand. Here's the active site, and there's the active site. So now you get it. It's kind of a flip like that. Flip around the back of the enzyme. But they did this so that you can see the nucleotides. And these are the four mutations, the four nucleotides that will most commonly change. And changing these to particular they're 4 amino acids. Yeah. Changing these 4 amino acids to specific other amino acids will allow the reverse transcriptase to work fairly well, although there is some fitness cost, and also be resistant to AZT. So, yeah. So AZT, a true antiviral. Maybe not the best one, but not a terrible one, at least in terms of effectiveness. Yeah. All right, so that's the only nucleoside reverse transcriptase inhibitor we're going to cover. There are many different ones. They mimic, all the nucleotides. And there are versions of those that can work against HIV. Yeah. And to make them, chemists essentially start with something that looks like a nucleotide, and they tinker. Yeah, relentlessly tinker. Sometimes they'll try to dock, like in a computer, their molecule onto the enzyme they're trying to inhibit, and see if they can get more contacts, a little bit better binding. And it's called rational drug design, and it's pretty neat. Okay, next class are called non-nucleoside reverse transcriptase inhibitors. So whereas a nucleoside reverse transcriptase inhibitor looks like a nucleotide, so here's our nucleoside analog, and here's our actual nucleoside or nucleotide once it gets its phosphates on. You can see they both got this ribose ring, although we've got this weird azide group down there. And we got the same actual base or something very similar up there. So yeah, non-nucleosides don't look like a nucleotide. They are just molecules that happen to fit into reverse transcriptase, and so everything that's not nucleotide looking and binds to reverse transcriptase is a non-nucleoside reverse transcriptase inhibitor. Now, turns out all the major ones bind to a little pocket. And this little pocket is kind of next to the palm. It's sort of in the connector region. And this, if I remember correctly, is kind of the hinge area that controls how the thumb and fingers are able to move. And these NNRTIs, non-nucleoside reverse transcriptase inhibitors, tend to bind that particular pocket. And so you can see this is actually a fairly symmetrical molecule. And so, yeah, a lot of the inhibitors tend to also be sort of symmetrical or have a kind of a symmetrical center on them. Yeah. Problem here is that you do get resistance. Resistance is by changing one or more atoms around this area. The HIV reverse transcriptase pays a little penalty, but not a very big one, which again is telling you that, ooh, if it can get resistance and only pay a little price for it, it's going to get resistance pretty quickly, and you're going to be in trouble. And what else? I don't know. Yeah. When you get resistance to one NNRTI, it tends to go along with resistance to several NNRTIs because they're binding the same spot. And when you make a change that basically is just like a little can opener, it pops, one of these nevirapines out, it'll also pop several of these other things out. And so, yeah, essentially you end up with just a resistant virus. So, not the best. All right. Next, antiviral, because why not? So the next class of antivirals are going to be integrase inhibitors. Our example is going to be raltegravir, although dolutegravir is also used pretty widely. You can see the oxygens on here. These are going to bind to the magnesiums at the active site, because remember there's an active site in the catalytic subunit of integrase. And you remember it's going to bind magnesiums, or it could bind manganese. They would bind in roughly the same way. And so, yeah, you're getting in there and interacting with the molecule in a way that prevents it from interacting with its normal substrate. And so it's just a little block. It looks like the thing the enzyme would like to pick up, but it's sort of deceptive. And the beautiful thing here is that your cells do not have, as far as I'm aware, anything closely related to an integrase, except in your endogenous retroviruses, and you don't need their integrases to work. So you can carpet bomb integrases, and you will get very nice effects. And there's no obvious mechanism for how you would end up with side effects. So, yeah, tends to be a good way to inhibit the virus. And the integrase gives you two ways to hit the virus, because of course the integrase is involved in integration, but it's also involved in assembly. The integrase is going to help us count 2 and only 2 genomes to put in, and it's going to make sure that the genomes end up inside that little capsid. Because when you put these integrase inhibitors in, sometimes you'll end up with a situation like this, where you get RNA that ends up outside the capsid. So it'll still get into the virion, but it'll end up outside the capsid, misassembled. And then when this gets put into the cytoplasm, you'll just have naked RNA floating around in the cytoplasm, which will probably be detected quickly and will probably be shredded before reverse transcriptase can do anything. So this is like an extreme example of what TRIM5 is trying to do with causing premature uncoding. This is, the code never formed properly. And so that's another thing that reverse transcriptase inhibitors can do for us, which is very nice. All right. Well, do we get resistance? Of course we get resistance. Integrase inhibitors are very useful. And yeah, you do unfortunately get some resistance. And the resistance is in these red amino acids. So these are showing you the ones that can change. And you can see these are the ones that are pretty close to this binding site. Oh, pardon me. And so the change you tend to see is that we will change this so that the drug does not bind as well, or so that the drug is blocked sterically from being able to bind. Yeah. Again, it's not rocket science. It's antiviral drug design. Okay, next one. So that was raltegravir. That was our integrase inhibitor example. Next one. Let's go for leptomycin B. This one I think is still in development. I don't think this is widely used by anybody as an antiviral, but it's got a neat mechanism because it's a Rev inhibitor. So after the virus has already gone into the nucleus, been integrated, and it's coming back out, you remember that Rev is going to get made from a spliced transcript, and Rev protein will then be translated, will come back into the nucleus, and I think by binding important, and once, yeah, there you go, important, and then once it is in the nucleus, it's going to bind to the Rev responsive element, the RRE, and then it's going to bind to CRIM1, and it's going to get shuttled right back out, and so we're going to take unspliced transcripts right back out of the nucleus. And the neat thing is that if you put in leptomycin B or one of these other experimental inhibitors, you can actually bind into this little pocket, which is very important for Rev interacting with one or more of the proteins that it has to interact with. And so, yeah, you stop Rev function, or you hold it back. And so, once again, you're giving the virus a problem, and you're forcing it to change something that the virus would be very happy not changing, because it has worked very hard to evolve a Rev that works very nicely, yeah. And so, yeah, frankly, it's kind of a jerk move of you to take that away from it, yeah. What are you thinking? You with your antivirals, yeah. All right, next kind of antiviral, and whatever, we'll talk Beviramat. This is from the company's website that makes it. They'd really like you to, be all on board with Beviramat. I don't believe this one is widely used yet either. It's more an investigational drug. Beviramat's thing is that it's going to bind to the GAG protein before it gets cut, and when it does, it actually binds, and it is going to bind in a way that prevents cleavage between the nucleocapsid part and the capsid part. So it's going to bind this little chunk that's sometimes called SP1. And by just sticking to that and covering over the cleavage site, now you don't get capsid processing. And so you end up with a virus with a weird looking messed up capsid that either didn't get cleaved right or didn't get cleaved at all. And so you're going to end up with something like premature uncoating. You're going to end up with a capsid that doesn't fit through the nuclear pore, possibly. It's just, yeah, you're just making a mess, a structural mess for the virus. And so that's great. That's another way we can win. And it's nice to see all of these things being used. Yeah. Okay. And then the last of our antiviral classes are going to be protease inhibitors. And so our example of a protease inhibitor is going to be saquinavir, which is on the next slide. There is saquinavir, and we'll come back. So this is the HIV protease, and this is the quizzical bulldog conformation. So it's the bulldog, but it's tipped its head to the side as if it's listening to you and understanding every word that you say. Yes. Do dogs do that? I don't know. They tilt their heads. That's all I know. And so what this does is it's going to bind to the active site, which the protease is actually a dimer. So each side of this is essentially one copy of the protease, and you need two of them coming together to form the full little bulldog. Yeah. And you remember that HIV protease is involved in maturation of the virus. And sure enough, so what saquinavir, how it was made is they took the literal site that HIV would like to be cleaving, and it cleaves in a conserved asparagine tyrosine. So asparagine would be N and tyrosine would be Y, and then the cleavage site would be right after that, just before this proline. And so we're taking that center, so it's going to bind perfectly, and we're just putting on two extra groups that will interact with the different parts of the protease that are already there. And so you're trying to make something that is basically competing with GAG for the right to sit at the protease. And there are some of these that will bind in there and not be cleaved because they'll just sort of block the protease from, moving and doing its cleavage. And there are others that will actually bind irreversibly to the protease, which is kind of neat, at the active site. So basically the protease will try to do the cleavage, and it'll end up with it's not a sequinavir, there are other drugs, but it'll end up with a protease inhibitor attached at the active site. And from that point, the protease is dead. Yeah, it cannot do anything. And so what you can do with a protease inhibitor is, even if you lost at every stage, so the virus got in, your Maraviroc didn't work, your T20 or Enfuvirtide did not work, the virus is in. Your nucleoside reverse transcriptase inhibitors didn't work. Your non-nucleoside RT inhibitors didn't work. Your integrase inhibitors failed completely. Your Rev inhibitors didn't even really try. Your GAG inhibitors missed. Somehow they whiffed on the target. You can still inhibit that protease, and you can still block maturation of the virus. And so, protease inhibitors. They tend to be used in almost all of the antiviral formulations. So, shown here are ways that the protease can change. And the primary mutations, these are mutations that will actually directly cause resistance. And so that's what the red circles are. The blue circles on the outside are other changes that the virus has to make, because when it changes this thing, it's changing a working part of a hard-working enzyme, and you can't just change this and not change the things that it sticks to. It's like if you have a 57 Chevy or something, and you try to cram the equivalent part for a 97 Toyota pickup into it, and you don't change anything else. It's not going to fit. You're going to have a mess, and why would you do that? Why would you do that? Yeah. These are what we would also call compensating mutations. And by by forcing the virus to make this change that it doesn't want to make, the red change, it also has to make the blue change, which it also doesn't want to make, just in order to keep working. And you've pushed the virus not just one step, but kind of two steps down the hill. And so, protease inhibitors, they can work, even though the only time they can work is after that particular cell has already lost at every step of the process. And HIV is basically won, but it's a final little, kind of middle finger to the virus. Say, yeah, you won this round, but you can't mature. That's too bad. Yeah, there you go. All right, and last thing we're talking about are antivirals. And so antivirals tend to be used in groups of 3 or occasionally groups of 4. And let's see. These are some of the main antivirals that are currently used. This is currently compliant, but we'll have to go back and look at this again every year. So, for example, a common what they call antiviral cocktail or antiretroviral therapy would have, dolutegravir. So let's look on our list. Where's dolutegravir? And you say, it ends like raltegravir. The A-vir part says it's an antiviral. The tegra says it's an integrase. Tegrase, yeah. And so a lot of antivirals that work against the integrase end in tegravir. Yeah. They don't have to, because again, those marketing whiz kids sometimes just say, no, we've got the best name. Yeah, we're going to call it Bob. Yeah, Bob the drug. What's it mean? Nothing. Is it going to sell? Like hotcakes. Yeah. You could pair that up. It's like a wine pairing. Your dolutegravir with your abacavir, or abacavir, I don't know, which is going to be one of these nucleotide reverse transcriptase inhibitors. And lamivudine is another one of these nucleotide reverse transcriptase inhibitors. But if I remember correctly, abacavir and lamivudine are mimicking different nucleotides. And so you can end up with these two things hitting the reverse transcriptase. And the cool thing is the changes it makes to become resistant to one actually don't help it much with resistance to the other. It's got to make additional changes. And so you're ending up with a active site of reverse transcriptase that's just all out of whack. Now, you might ask, well, why don't we put like 10, 15 different, inhibitors in the same cocktail and use them together? It's a good question. Theoretically, it can work. You would have a larger pill, and you would have a more expensive pill. And in terms of practical concerns, what you tend to get are all of the drugs developed by one pharmaceutical company packaged together in one convenient little pill. And so a company will tend to put their protease inhibitor with their reverse transcriptase inhibitor with their NRTI into a nice little pill, and then another company will do the same thing with their versions of them. They kind of work in a similar way, and they can both be effective. But yeah, that's it. Yeah. And so that is sort of current antiviral therapy. So if you were asked on a test of some sort, can't think what kind, to come up with an antiviral cocktail, this would be the way to do it. So you'd want to look for, and I'd say the easiest way would be to look for antivirals that hit 3 different parts. And it would be totally okay to say even antivirals that are still in development or something like that. That would totally be fine. So in addition to the ones that we talked about, there are also strand transfer inhibitors. Elvitegravir is one of those. And there is also a group called cytochrome P450 inhibitors. So cytochrome P450 or CYP450 enzymes are found, in a lot of cells, but most predominantly or famously in the liver. And these are involved in the breakdown of antivirals. And so by inhibiting them while your antivirals are present, you can get more effect out of your antivirals by essentially slowing down the part of the host that would be otherwise breaking down those antivirals. There you go. And so you can see it's just in one of these combinations, cobicistat. Yeah, it's right there. And there you go. So that's it. That's HIV antivirals, or at least that's enough of HIV antivirals to get you going and get you exposed to the subject. So thanks very much.